Today I would like to talk about APOSEC, a possible new therapeutic option or treatment for stroke and spinal cord injury. I would like to start off by talking about our experiments conducted in the stroke preclinical model in rats. First off, to stroke and some general information and why it's still worth to try to explore a new type of treatment. First off, stroke is a really common disease. It accounts for around 9% of deaths worldwide each year. And this number will even increase within the next couple of years due to the demographic development and lifestyle factors. Talking about the etiology of stroke, we have to say that around 90% of strokes are caused by ischemia. It means that some kind of thromboembolic event results in occlusion of a vessel, and the supplied brain tissue is then impaired in its function, leading to neurological impairment. The golden standard or the current treatment option is to resolve the thrombus and try to restore blood supply to the impaired brain tissue, and this is accomplished by thrombolytic therapy. However, only 6% of patients with ischemic strokes are eligible for this treatment due to contraindications like anticoagulation therapy. And therefore, there's still a need for multi-layered strategy, targeting not only the revascularization, but also other important steps within the progression of stroke. On the next slide, you can see the different parts of the pathogenic mechanisms going on after an ischemic event. You can see, indicated by the thunderbolt, that this ischemia starts a cascade including inflammation, energy deficit, oxidative stress, excitotoxicity, and apoptosis. And all these mechanisms lead to further damage to the brain tissue. Another fact is that after a stroke or an ischemic event, not the whole brain tissue, accounting for the neurological impairment, is structurally lesioned at the beginning. There is a so-called penumbra surrounding the structural lesion site, which is potentially salvageable and can be saved by, on the one hand, restoring blood supply, but also by other mechanisms. However, if this ischemia holds on for a period of time, the structural lesion will continue or will grow and lead to a lesion site that is not salvageable anymore. We, in our group, tried to investigate the characteristics and therapeutic potentials of the secretome obtained from apoptotic peripheral blood mononuclear cells, and we term this as APOSEC. It's an acronym of the secretome obtained from apoptotic PBMCs. The next slide shows you how we produce this secretome. We start off by performing a venous blood withdrawal from healthy probants. Then we separate the PBMCs by a density centrifugation protocol. We then irradiate these cells and therefore triggering apoptosis. Then we incubate these cells for 24 hours. And after this incubation period, we centrifuge the cells and discard the cells and just use the secretome for further processing. Then we lyophilize the secretome. And this lyophilized secretome can be easily stored. And we add another virus elimination step in order to meet GMP guidelines. We were able to show within the last couple of years that the secretome obtained from apoptotic PBMCs, or how we call it APOSEC, is able to regenerate ischemic myocardium, inhibits platelet activation, but also reduces microvascular obstruction. And therefore we were interested if these therapeutic characteristics were also applicable in a stroke model in rats. We utilized a suture model called MCAO, where a defined suture is inserted to occlude the middle cerebral artery. Here you can see on the right-hand side the situs after dissecting the carotid artery in rats and the bifurcation into the internal and the external carotid artery. In the next step, you insert in a stump created of the external carotid artery. You insert the suture until a resistance is reached, and the defined diameter assures that you reach the middle cerebral artery. And this leads to an ongoing occlusion of the middle cerebral artery and therefore an ischemia in this perfused area. This can be visualized by utilizing the TTC staining. This is a molecule that is reduced in vital tissue and therefore stains the brain tissue in red. You can see on the right-hand side all the white area that is not stained by reduction of TTC remains white and is therefore dead. And this accords to, or is in accordance to, the perfusion area of the middle cerebral artery. This can also be used to quantify the extent of the ischemic area. Now on to the results. First off, we were interested if incubation with our secretome is able to induce different pathways in vitro. We therefore took primary human astrocytes and Schwann cells, as indicated by AC and SC on this figure. Then we took a control medium and APOSEC and incubated these cells, and then performed immunoblots to investigate the phosphorylation status of different proteins. And we found upregulations of several pathways, and these pathways now marked in red are very important in the progression of stroke events. For example, the CREB pathway, also the ERK1 and 2 pathway, but also UN, ACT, and HSP27. We further found a dose-dependent upregulation or phosphorylation of CREB in astrocytes and also neurons, which is indicated in this figure. You can see that starting with 0.25 times 10 times 6 and going up to 25 times 10 to the 6th. And if we go back to this figure I showed previously, you can see that these pathways are important in the apoptosis and progression of damage after stroke events. And these pathways, if upregulated, are able to attenuate damage following stroke. We then were interested if these upregulated pathways are also able to induce functional changes, and were able to find increased neuronal sprouting in an in vitro sprouting assay. You can see on the control slide the sprouting going on with just the medium applied, but on the lower picture you see that after incubating neurons with the secretome, increased sprouting arises. And this is also depicted on the right-hand side, indicated by the red bar, and this was significant as well. Now I want to talk a little bit about BDNF. BDNF is the brain-derived neurotrophic factor, and it's a member of the neurotrophin family of growth factors, which were largely investigated during the last couple of years, also in the stroke setting. Here you can see a figure from a very recent paper, and they administered BDNF into rats after inflicting these rats with stroke. And they were able to show that the application of BDNF alone helped rats recover better from neurological impairment following stroke. You can see on the left-hand side the beam walking test performance score, for example, whereas a lower score would mean they make less errors and perform better on the test. And you can see that here that after administering BDNF, the mice recovered better in all the tests. Therefore, we were interested if BDNF is also present in our mix of soluble factors in APOSEC. And therefore, we performed ELISA analysis and found that indeed BDNF is present in this mix of factors. The other neurotrophic factors were not present, for example, glial-derived neurotrophic factor or neuronal growth factor. but BDNF was present also to a large extent. And then we want to ask if also administering of APOSEC in vivo could lead to an upregulation of endogenous BDNF production. Therefore, we injected APOSEC intraperitoneally into rats and found that this administration led to higher BDNF plasma levels. Important to mention is that we used a specific rat ELISA for this experiment, ensuring that this BDNF was indeed newly secreted BDNF in the plasma. Now we went on to the in vivo model of stroke, and we saw that treatment of APOSEC led to a reduction of infarct volume, and our treatment, both the control group and the treatment group, received their agent intraperitoneally. So this is a systemic effect we are seeing here. And we further wanted to see if this reduction of infarct volume also leads to some kind of improvement in neurologic function. Therefore, we evaluated the neuroscore. The neuroscore goes from 0 up to 7, while 0 means no impairment observable at all, and 7 equals death. So the lower your score, the better you perform neurologically. And you can see here indicated with the red line that animals treated with APOSEC recovered better within the first 48 hours after middle cerebral artery occlusion. Now, before discussing results obtained from the stroke experiment, I would like to continue with results obtained from our second set of experiments, from experiments obtained from spinal cord injury model. First off, again, some facts about spinal cord injuries. We have an incidence of around 50 per 1 million each year worldwide. And a big problem considering spinal cord injuries is that mainly young patients are affected, with the mean average of trauma being 31 years. This leads to a long period or long lifetime with a handicap. And this handicap is often really dramatic in terms of neurological impairment. Around 50% of patients suffer from total loss of motor function below the level of lesion. This is further aggravated by the fact that many patients, or most patients, 2/3 of the patients, suffer from spinal cord injury at the level of the cervical spine, also due to the fact of the highest mobility within this region. So we have a really high level of lesion and often tetraplegia occurring in these patients. Furthermore, many patients, around 70%, suffer from chronic neuropathic pain, a pain that is characteristically difficult to treat and leads to big clinical problems. Here depicted you can see the pathomechanisms going on after spinal cord injury. After a primary injury caused by some kind of mechanical impact, in most cases motor vehicle accidents, and afterwards there's a secondary cascade that kicks in and aggravates the damage and leads to more severe neurological impairment. And this is exactly the process or the mechanisms most therapy regimes try to intervene and try to dampen the aggravation after the primary injury. And we also believe that our secretome could be important within the modulation of this secondary process. Here on the right hand you can see the secondary process going on in detail. It's a vicious circle, and it starts with vascular disruption and ischemia, continues with inflammation, production of free radicals, lipid peroxidation and excitotoxicity, and this closes again the vicious circle, leading to even more damage and necrotic as well as apoptotic death of neurons and increased disability. Therefore, the aim of our study was to investigate possible therapeutic effects of APOSEC in a spinal cord injury preclinical model, and therefore we used a contusion trauma model utilizing this impactor you can see on the right top picture. What you have to do is you perform a laminectomy under the microscope. We chose the level of the 11th thoracic vertebra, and then you apply a force with this impactor onto the exposed spinal cord. And afterwards we performed BBB score analysis to evaluate hind limb function. I will go into this in more detail later on. And we performed histology on day 3 and day 28 after the trauma. Now here on to the results. First off, we started with ex vivo experiments. We used an aortic ring assay where we took rat aortic rings and incubated them with either control or secretome with APOSEC. And we were able to show that APOSEC exhibits a vast extent of angiogenic potential, as you can see here by the sprouting activity going on the right picture. We then did control experiments because we wanted to make sure that not only VEGF in this mix of soluble factors is responsible for the effect we're observing. Therefore, we added a neutralizing antibody against VEGF and saw that there is still angiogenic potential available in the secretome. We then did further control experiments with recombinant VEGF and recombinant VEGF plus neutralizing antibody just to confirm our results. And we further quantified these observed sprouting activity, as you can see now on the right hand side. Because we were interested in therapeutic potential in central nervous system structures, in spinal cord and previously in stroke, in brain tissue, we therefore asked the question if these ex vivo results we were getting from the aortic ring assay are still applicable if we use central nervous system tissue. Therefore, we developed a new assay where we took the same steps as in the aortic ring assays, besides of using spinal cord tissue instead of aortic rings obtained from rats. And after incubation of these spinal cord tissue samples with control medium or APOSEC, we're still able to see this sprouting activity because of our secretome. And this was also quantified, as you can see now on the right hand side. But now the question arose if this really is vascular structure we're seeing sprouting out. Therefore, we added an antibody against endothelial or endothelial marker and performed confocal laser microscopy, and were indeed able to see newly forming vessels indicated as the green lines of newly formed endothelial layers. In blue, you can see nuclei stained with DAPI. Then we went on to in vivo experiments. We administered APOSEC intraperitoneally and measured cytokine levels in the plasma of the rats. And we were able to see a significant increase of CXCL1 12 hours after administration. CXCL1 is known also for its angiogenic potential. And we saw that after 12 hours again, this level started to decrease again, indicating that this was really triggered by our administration of APOSEC. The other factors measured, interleukin-10, TNF-alpha or interleukin-1 beta, weren't detectable during this experiment. Now on to results obtained from the spinal cord injury model. Here you can see a histology cross-section of the spinal cord 28 days after trauma. And you can see here a large cavity formation in the dorsal column of the spinal cord. This is a control animal, and this cavity formation was not present in animals treated with APOSEC, which is also indicated on the right-hand side, where this cavity formation was quantified. We further were interested in axonal pathology within the spinal cord. On top you can see the animals treated with the control. On the lower picture and picture below you can see animals treated with APOSEC. And we performed a beta amyloid precursor protein staining. And this is a protein ubiquitously expressed in the central nervous system. And this protein also undergoes heavy axonal transport. And if the axon is damaged, the amyloid precursor protein will accumulate and therefore will be visible within this staining. And as you can see on the top picture, there are many brownish dots indicating the accumulation of beta amyloid precursor protein, and this was not present in animals treated with APOSEC. On the right-hand side, again, quantification of these analysis. The next question we asked was whether APOSEC is able to modulate the inflammatory response following spinal cord injury. On the left-hand side, you can see spinal cord tissue from animals with control treatment, and on the right-hand side with APOSEC treatment. And indicated by the white bar on the right-hand side, you can see that APOSEC-treated animals experience a high number of CD68 positive cells within their spinal cord 3 days after trauma. Now the problem with CD68 staining in the central nervous system is that there are resident microglia cells present which are also capable of expressing CD68. And there is no known marker so far for distinguishing between these two cell types. Therefore we performed a second staining against the inducible nitric oxide synthetase, short INOS, and this serves as some kind of surrogate parameter indicating oxidative stress. On the left-hand side you can again see medium controlled animals, on the right-hand side Secretome treated animals, and you can see that despite the upregulation of CD68 expression in treated animals, there is lower amounts of inducible nitric oxidase synthetase levels present, indicating that these cells are not pro-inflammatory microglia, but rather are monocyte-derived macrophages. And this is important because of this study published in 2009 in PLOS Medicine, and the authors were able to show that these cells, namely monocyte-derived macrophages, are pivotal for the recovery after spinal cord injury. And this is depicted in this figure right here. You can see on the left-hand side a motor score, and it starts with 1, indicating no observable hind limb function, and ends with 9, meaning totally perfect hind limb function, so no impairment at all. And what they did was they took a conditional knockout mouse model where they were able to knock out or get rid of all the monocytes, and this group is indicated by the red line, where you can see that this group experience no recovery after spinal cord at all. If you extinguish the monocytes and after replenish them with adding monocytes again, you can regain the recovery observed in the control group as well, indicated by the bright green line. Here you can see the cross-section of a control animal 28 days after trauma, and this is again a CD68 positive, a CD68 staining. And what we know from other publications is that 28 days after trauma, only microglia is remaining. Monocyte-derived macrophages underwent apoptosis and are not present anymore in the spinal cord. And here you can see still activation of microglia going on in the white matter of the spinal cord, indicated by the brown staining. And this staining was not so highly positive in our APOSEC-treated animals, suggesting a higher degree of resolution of the inflammation 28 days after trauma. Now we performed another staining. We stained against the von Willebrand factor to stain for vessels, and we were able to show that animals treated with APOSEC showed a higher density of vessels in the spinal cord 28 days after trauma, as indicated by the arrows, and this was also significantly higher after quantification. Now, the most important question is whether these changes we observe are also able to result in neurological improvement and increase or improve the outcome. And here you can see the BBB score. It's slightly different from the score I've shown you before. It's a score specifically for rats. And it starts with 0, indicating no observable hind limb function. It goes up to 21, meaning no observable impairment at all. So perfectly fine functioning hind limbs. And you can see, meaning that the higher your score is, the better your hind limbs work. Here you can see that APOSEC treated animals improved faster and better than control animals, and this was significantly higher starting on day 14 and remained significantly during the experiment until day 28. Now I would like to discuss what I just talked about. First off, we talked about secretomes of apoptotic PBMCs, and in previous studies were able to show that there are several characteristics that might have beneficial effects also in this setting of stroke and spinal cord injury, namely cytoprotection, immunosuppression, and also inhibition of microvascular obstruction and induction of vasodilation. And with this set of experiments, we were able to show that these properties are indeed able to have beneficial effects or therapeutic effects in a preclinical stroke model or and as well in a preclinical spinal cord injury model. We suggest that the mechanisms involved in this recovery or in this improved recovery after treatment are due to angiogenesis observed in the aortic ring assay, but also in the spinal cord tissue assay, suggesting that this effect is still present and available in central nervous system. Further on, inhibition of microvascular obstruction, which leads to reduced vessel degradation, which can be observed within the higher vessel density in our histology evaluations. Furthermore, we were able to show that the inflammatory response is being modulated by our secretome, leading to a modulation of the inflammatory response in terms of enhancing monocyte recruitment to the spinal cord, but dampen the microglia activation. Furthermore, we saw upregulation of intracellular pathways associated with neuronal surviving, but also with increased sprouting in vitro assays. Furthermore, we found levels of BDNF, of brain-derived neurotrophic factor, in the secretome itself. And the secretome is further able to upregulate BDNF in vivo, leading to a higher secretion in rats after administration. This all can account for a multi-layered therapy, which is needed because of the low availability of patients who are available for the revascularization therapy. Now I would like to conclude. And with this set of experiments, we suggest that secretome of apoptotic peripheral blood mononuclear cells, APOSEC, provides an interesting therapy option for upcoming trials investigating stroke and spinal cord injury. And with this slide, I would like to end and thank all the people for helping realizing these experiments and you for your attention.